I would like to thank you for the kind opportunity to present our work here, especially for the organizers. It's a great honor for me. I'm representing Professor Peter Knight today from the National Institute of Oncology and his team. And what I am going to talk about is a little bit different from the previous presentations, as what we do is basic research with a potential translational future. So the story I'm presenting today is our recently published cell metabolism paper. This work was dedicated to investigate how targeted therapy resistance evolves in melanoma, and especially with a metabolic point of view. So B-raf V600E is present in approximately 50% of skin melanomas. It leads to an overactivated MAPK-ERK pathway and therefore to non-controlled proliferation. This is basically a RAS-independent process. The first targeted therapeutic options were introduced approximately 15 years ago. They were really promising, but unfortunately resistance quickly developed. Further studies suggested that in most melanomas resistant to B-raf inhibitors, MAC kinase is overactivated, and therefore this led to the introduction of dual therapies when next to B-raf, the MAC kinase is also inhibited. This led to better therapeutic response, but unfortunately resistance remains a problem, even with the nowadays available immune therapies as well. Our understanding of therapy resistance developed in the past couple of years. As previously we thought that resistance is mostly a genetic process, which is true and which is extremely important, but now we know that there are additional layers of therapy resistance that make it more complex, but possibly they could also lead to new targets. So from a therapeutic point of view, if we consider a cell population, there are the therapy-sensitive cells, those die during the therapy. There are the resistant cells, which have initial mutations causing them to resist the therapy. And there is another cell population which are really interesting, and they were discovered in the past few years. These are called the drug-tolerant persistor cells. The interesting thing about them is that they survive the therapy, they remain in a quiescent or slowly proliferating state, but the changes in them are transient, reversible, and only phenotypic. And this is the interesting thing about them. But after targeted therapy continues, they could gain another mutations, and they can evolve and acquire resistance. So the whole aim of our study was to characterize the persistor and the resistant cells and find out which changes are present in the persistor cells and which remain there in the resistant cells with the aim to find new potential targets. Here you can see that these are, we did an in vitro model using B-raf mutant melanoma cell lines. Here you can see that persistor cells do not proliferate in the early stages, but resistant cells almost regain their proliferative activities, similarly to therapy-naive cells. An interesting thing about the resistant cells that they gain PI3 kinase AKT overactivation, which is very common in MAPK inhibitor-resistant tumors. So there were previous studies that suggested that oxidative stress is a common thing in melanomas resistant to MAPK inhibition. Our aim was to find out what the situation is with persistor cells, and we also found that persistor cells are also characterized with increased oxidative stress, and this manifests on the level of protein oxidation and also in poly-ADP ribosylation, which is an indicator of oxidative stress-induced DNA damage. When we were trying to find out the sources of ROS, I mean reactive oxygen species, we found that cytochrome P450's family of proteins involved in the neutralization of drugs which act as monooxygenases producing reactive species were overexpressed in persistor and in resistant cells. And we also found that both persistor and resistant cells are characterized with increased mitochondrial respiration, oxidative phosphorylation, which is another important source of reactive oxygen species-induced oxidative stress. When we characterized these cells metabolically, we found that persistor cells suppress glycolysis, whereas resistant cells regain their glycolytic capacity, which is in line with the fact that persistor cells do not proliferate, and highly and quickly proliferating melanoma cells mostly need glycolysis for the operation of high proliferation. When we were investigating the antioxidant properties of these cells, we found that persistor cells overexpress a high amount of different kinds of antioxidant proteins. In resistant cells, especially this thyrodoxine reductase was overexpressed. So this means that these cells react to the oxidative stress with the arsenal of these proteins. When we checked the regulatory proteins which regulate the flux, the metabolic flux between glycolysis and then the pentose phosphate pathway, in accordance with these, we found that persistor cells activate the pentose phosphate pathway to produce an NADPH, which is needed by these systems, which lead to redox homeostasis and cell survival. Whereas in the therapy-naive cells and the resistant cells that need glycolysis to proliferate, the flux is shifted back, which is in accordance with our seahorse measurements. One of the most important amino acids for us redox biologists is cysteine. This amino acid is used for redox regulation. It's a building block of different cofactors, redox molecules, and also redox activated proteins. And this amino acid is mostly taken up by the cells from the extracellular space in the form of cysteine. It's in oxidized form. And when we checked, we found that both persistor and resistant melanoma cells take up more cysteine, which is also in accordance with the increased antioxidant demand. And interestingly, when we combined the MAPK inhibitors with the inhibitor of this transporter, we found in vitro that the resistance to therapy could be delayed, also suggesting that cysteine is really important for these cells to survive. Most of the cysteine in the cells is utilized for glutathione synthesis. This is basically an antioxidant molecule. It's a tripeptide, and it's present in high concentration intracellularly. And here we found that it's not only, it doesn't only act as a redox buffer, but it can be also conjugated to different synabiotics by these GST enzymes, which were also overexpressed in persistent and resistant cells. So this means that a huge amount of cysteine is used for glutathione synthesis, which is used to counteract the oxidative stress, but also to neutralize the drugs or to have therapy resistance to evolve. Cysteine is not the only limiting factor for glutathione synthesis. Another limiting factor is glutamate, the amino acid glutamate, which is also used for cysteine uptake, but also used for citric acid cycle, which produces NADH for the increased oxidative phosphorylation. So when we investigated glutamine and glutamate metabolism, we found that both persistent and resistant cells need more glutamine to make more glutamate, which is, it's an interesting thing that the persistent and the resistant cells use different approaches. The persistor cells overexpress the glutaminase, so they make more glutamate from glutamine, whereas the resistant cells simply suppress glutamine synthase, so they don't utilize glutamate to make glutamine. But the bottom line is that both cell lines under therapy need glutamate to make glutathione and to take up more cysteine, and also to use it for the activation of the citric acid cycle and therefore to oxidative phosphorylation. Cysteine is an interesting amino acid, as upon extracellular cysteine limitation, it can be de novo synthesized from methionine by the transsulfuration enzymes on the canonical pathway. And these enzymes are the CBS and the CSE, and both of them are needed to produce cysteine. So we checked this pathway and saw a very interesting pattern, as we saw that persistor cells overexpressed CSE but suppressed CBS, and the resistant cells showed a similar phenotype to the therapy-naive control cells. The interesting thing about this is that both enzymes are needed to make cysteine, so this way it doesn't help them to make it. But these enzymes have another role, which are independent from each other, and not just independent, they can compensate each other's role to make these reactive sulfur species. Reactive sulfur species are less widely known. They are basically sulfur-containing reactive molecules. For example, hydrogen sulfide is more widely known as it is responsible for the bad smell of the rotten egg, but it's also a very important antioxidant small molecule. And persulfides are even less well known. These are similar to thiols, but they contain an SS bond, and this makes them highly reactive and also very good antioxidant. And both of these molecules are also important in cellular signaling events. And how persulfides protect the cells is basically that when oxidative stress occurs, these persulfide species can be reduced back to native thiols, whereas normal thiols that undergo huge overoxidation are determined to degradation and therefore to loss of function. So when we investigated the levels of reactive sulfur species, not surprisingly, we found that most of these species were elevated in persistor and resistant cells, although their levels are usually tightly regulated. So we thought that this might be important in persistor cell survival. To show this in vivo, we used a cell line derived and also a patient derived xenograft model. And we were able to show that these changes we saw in vitro were also present in vivo regarding cysteine metabolism and also the production of these reactive sulfur species. And of course, as I showed you, CSE was overexpressed, so in this case we focused on CSE, and we asked whether this reaction could be the major source of reactive sulfur species production. And to check this, we used different genetically modified melanoma cell lines, and we were able to show that indeed CSE is important in persulfide production, which could help persistor cells to survive. When we check the levels of protein persulfidation, which is not only an important protector, but also has implications in cellular signaling, as it is also a post-translational modification, similarly to protein phosphorylation. And we saw that especially in persistor cells, protein persulfidation is elevated, and there are bands where the proteins are especially elevated. We are still working on that to identify these proteins using a proteomics approach. We were able to show it with, for example, this PRX2, that it's happening. To show whether persulfidation is indeed important for persistor cell survival, what we did was we silenced one of the genes coding a protein responsible for persulfide degradation, and therefore we kind of shifted the balance of degradation and production towards production, leading to an increase in persulfide levels. And we found that when these cells were treated with MAPK inhibitors, the silent cells started to proliferate earlier, which means that indeed increase in persulfide levels is adaptive for these cells to survive the therapy. I don't want to go into details, but another interesting thing about these reactive sulfur species that they are able to donate electrons to the electron transport chain, which could activate the oxidative phosphorylation even more. And I showed you on the beginning of the presentation that maximal respiration and respiratory capacity is increased upon MAPK inhibition, so we think that reactive sulfur species donated electrons could contribute to this in addition to an ADH. And why is this important? Is because we also found that a very important transporter, ABCG2, which is responsible for drug efflux, is highly overexpressed in especially in resistant cells, and these transporters are very energy demanding. So we think that the high energy demand of cells must be met via these metabolic reprogramming. Of course, our major goal was to identify new targets, and for that we asked whether CSE could be a good target. To test whether there is any physiological implication, thanks to Professor Liszkai, we were able to find a few samples from the same patient with, before and during therapy. And although the number of the samples was low, so it's not, it cannot be used for statistical analysis, but we were able to show at a few paired samples that there is indeed an increase in CSE levels in the patient as well, although the levels of CSE were very different in different kinds of patients and also could depend on the site of the melanoma itself. The reason we think CSE is a good potential target is because it's selectively overexpressed in the persister cells. The other enzyme, CBS, which could compensate for its function, is suppressed, and also CSE has a relatively selective, commercially available inhibitor. In the next experiment, we used the CSE overexpressing cell lines we generated previously, and we found that if we elevate the levels of CSE even more, they are gaining resistance earlier. And if we use the selective inhibitor I mentioned, the propylglycine, PEG, shortly, we were able to suppress the persulfide production, which is very important, as we could show that either by genetical or by pharmacological inhibition of CSE, we were able to delay the onset of acquired resistance in vitro. The exact molecular mechanisms are not still clear for us. We are still investigating it using high throughput methods, but one thing about them could be that AKT phosphorylation, so the activity of PI3 kinase AKT pathway was lower in the PEG-treated cells, which could mean that there are some redox signaling implications in the background, but we are still working on this part. Most importantly, when used in vivo, CSE inhibitor, MAPK inhibitors combined with CSE inhibitors increased the progression-free survival of tumor-bearing mice, and basically this is the basis of our new patent application and our future efforts for drug development. The preclinical studies, the early preclinical studies just started last month. So just shortly summarizing our results is that using MAPK inhibitors, cells overexpress cytochrome P450s and activate oxidative phosphorylation, both leading to the production of reactive oxygen species, which is a stress for the cells. To survive this stress, cells take up more cysteine and overexpress CSE, producing reactive sulfur species, which also could enhance the activity of oxidative phosphorylation to produce more ATP for drug efflux. But also these reactive sulfur species are important in protein persulfidation, which leads to an elevated redox protection, an altered cellular signaling, therefore to persistor cell survival and on the long run acquired resistance. When we were inhibiting CSE activity either genetically or pharmacologically, we were able to delay the resistance in vitro and in vivo as well. I'm really happy to work in such a great team. I would like to say especially thanks to those who worked on this project in the upper line. And I would like to say special thanks to Professor Liszkai for the patient samples, for Dr. Erika Tóth for the immunohistochemical analysis, and this in vivo work wouldn't be possible without Dr. József Tóvári and Dr. Mihály Cserepes. I am very grateful for our founders and for the institution for their amazing support. And with that, I would like to thank you for your kind attention.